Hello, my name is Mark Temple, and this is a presentation on cancer genetics as part of the In Brief series. And in this presentation, I'll be looking at cellular oncogenes. So, by way of introduction, within the human genome, clearly there are thousands of genes, tens of thousands of genes. Some of these genes play really important roles in cell signaling. They play important roles in controlling how cells respond to growth factors and how cells mobilize gene expression to proliferate or differentiate or go through some large cellular process. Okay, so these important genes that are involved in these signaling pathways, often they can get mutated. And when these get mutated, they can signal to the cell to keep dividing when they shouldn't be dividing, or they can signal to grow in the absence of growth factors. So there's been a bunch of these key genes identified, and they're referred to as either oncogenes and tumor suppressor genes. In this short presentation, I'll be talking about oncogenes. I've done another presentation that talks about tumor suppressor genes. So the important thing about, as we'll see in this presentation, the important thing about oncogenes is that the coding sequence for these genes can pick up small mutations, single point mutations often, and these lead to hyperactivation of the gene. So if you've got two copies of the gene in the genome, one of them gets mutated. That mutated copy is hyperactive. So even though there's a normal copy there, you've got the hyperactive copy there. So only one mutation on one copy of the gene is required before you can see the effect of the mutation. Now, the poster boy for oncogenes, if you like, is the Hras gene. It was one of the first oncoproteins identified. An oncoprotein can be described as a proto-oncogene or an oncogene, or an oncoprotein, but oncogenes. So a proto-oncogene is the normal copy of the gene that's not mutated. That's a proto-oncogene. Onco just means, like, it's a reference to cancer, so it's a gene that can be involved in cancer. An oncogene, fully fledged oncogene, is the proto-oncogene that has been mutated. Okay? And I guess another thing I should say about oncogenes, an oncogene doesn't really talk about the function of the protein. An oncogene is a category of proteins that are important in cell signaling. Now, it may be that the oncogene is a receptor protein on the membrane of the cell. It may be that the oncogene is a switch protein in the cytosol. It may be that the oncogene is a transcription factor. Okay, so oncogene doesn't refer to the biological function. It just says that these important genes that are involved in signaling can get mutated and cause cells to be growing when they shouldn't be growing, or undergoing some process that shouldn't be occurring. Okay, so we'll look at the Hras gene as a typical example of, or at least a well understood example of an oncogene. Now, when they looked at the DNA sequence of the oncogene compared to the normal copy, the proto-oncogene, they noticed a single point mutation in the coding sequence. And this was a change in a guanosine to a thymidine. So the big change was that guanosine in one of the codons had been replaced by thymidine. Okay, you'd think it's such a small change, it's not going to have a big difference. So in this diagram here, this shows the coding sequence of the first exon, or at least part of the first exon, of the H-ras gene. So here you can see is the ATG codon. That is the start codon and codes for a methionine. And then there's groups of 3 bases, consecutive groups of 3 bases, each coding for a different amino acid residue in the protein. So, we've got tyrosine and lysine and leucine and valine and all of these amino acids. Now, along the sequence here, typically in the normal copy of the Hras gene, the proto-oncogene, there's a G residue here. And what's shown in this, on the bottom sequence, is the oncogene, the mutation in which the, as shown in the previous slide, the guanosine is replaced by a thymidine. So if you look here, the guanosine is replaced by a thymidine. Now, this has a change in the amino acid coding sequence. So if you look up your genetic code of codons to amino acid residues, the original sequence, which was the GGC, coded for glycine. Point mutation at G, we now have a T. And this GTC codes for valine. So what we've done, you've swapped the glycine residue in the normal copy of the protein for a valine residue in the mutated copy of the protein. Now, if you think about it, glycine is the shortest, smallest amino acid residue there is. It's just a hydrogen atom off the backbone. Valine is this big protruding methyl groups. It's a big like tree of an amino acid residue, as opposed to this tiny little glycine. So you put this big sort of block, this big bulky thing into the protein sequence here. And that causes a change in protein function, and it causes the Hras protein to be hyperactive. So as highlighted here, there's a point mutation causing a change in the codon, and the amino acid residue change results in glycine changing to valine. Now, how does that look in the actual protein? So shown here is a 3D structure of the Hras protein. And this is taken from another one of these presentations in cancer genetics. Now, shown in this part of the diagram here, you can see this thread here, this loop of the protein. And within this loop of the protein, there's is the glycine 12. So residue number 12 is shown here. And this residue number 12 is interacting, well, it's been replaced by valine, and you can see it's interacting with the bound nucleotide that's within the Hras protein. So one thing you need to know about the Hras protein is that it's a GTPase. It's a protein that binds a guanosine nucleotide. And this purple structure here is that nucleotide. Okay, so there's the base, the guanosine, and here's the triphosphate part of the guanosine, and you can see that these triphosphates on this nucleotide are interacting with the protein at the point where the mutation has occurred. And this part of the protein here is part of the effector loop, which signals which allows Hras to signal to other proteins to push the cell to grow. So this was a really important discovery in molecular biology in relation to cancer. It was the first time that a single point mutation had been discovered that contributes to the growth of a human cancer. So as soon as people understood the process, you could start looking for this mechanism in other genes, in other oncogenes, and in these tumor suppressor proteins as well. So, and another important point about this is that this point mutation occurred as a somatic mutation, meaning it didn't occur in the germ cells. It wasn't part of the hereditary profile of the family. It occurred in an adult cell as a point mutation in response to a normal cellular process, or a carcinogen, or sunlight, or smoking, or something had caused that point mutation in somatic cells. So once people knew that the Hras protein played an important role in cell signaling, and once it had been discovered that a mutation in this can lead to transformation of cells, then people started to look in cancer biopsies. And in the 1980s, it was demonstrated that up to 30% of solid tumors showed a mutation somewhere in the Hras gene. And certain positions in the Hras protein are clearly more sensitive to mutations than other regions, particularly if they're in that effector loop, in that nucleotide binding region. And the replacement of the glycine at position 12 with any other amino acid, except maybe for proline, leads to an increase in the tumor transforming potential of the Hras protein. So if you basically, gly is a really small amino acid residue. If you replace it with any other residue, all of them are bigger than glycine. All of them have got a side chain, and therefore all of them have an effect on the function of the Hras protein. So I've got a little animation here which shows the difference between Hras in the on state and the off state. So this is normal Hras, this is not the mutated Hras, and what it's showing here in this diagram is here is the nucleotide. So here's the guanine triphosphate, the GTP, with the three triphosphate groups here, and it's showing the interaction of the phosphate groups with this effector loop, including the glycine 12, which I imagine would be the small amino acid side chain here. And in this animation, it switches between the Hras in the on state and Hras in the off state. So they've taken the two structures on and off, and then they've sort of morphed the change that needs to occur to switch between one and the other, as shown here. So this is Hras in the on state, switching the effector loop to Hras in the off state. So what exactly is the Hras protein? It's referred to as a GTPase. And a GTPase has the ability to hydrolyze GTP to GDP. So the triphosphate is hydrolyzed into a diphosphate, and one phosphate group is removed. So when Hras is bound to GTP, it represents the active form of Hras. So when Hras is bound to GTP, the effector loop is exposed and it's signaling to the cell to turn on genes that are involved in processes such as proliferation. Okay? At some point, the Hras can hydrolyze the GTP to GDP. This allows the effector loop to then return into a different conformation, and this conformation with the bound GDP is inactive, and it stops the signaling to the nucleus to turn on gene expression. But the actual biological property of Hras, it's a GTPase. So it's the hydrolysis of that GTP that causes the conformational change. The conformational change either switches Hras on or off. And in any molecular textbook will have a diagram showing the activation and the turning off of the Hras protein. And I've done my own little animation here to represent the Hras activation cycle. So shown here is Hras in the active state bound to GTP, and I've represented the phosphates here as two red and one green circle. And I've drawn a little blob here on the active Hras protein, indicating that the effector loop is exposed and that it's able to turn on gene expression. Now, Hras is a GTPase, meaning it can hydrolyze the GTP to GTP. So during the hydrolysis here, one of the phosphates is removed, and that gives rise to Hras now in a different structure without the effector loop, bound to GDP. Now, the process of Hras hydrolyzing the GTP can be significantly enhanced by other proteins, which are referred to as gap proteins. So the gap proteins are GTPase activating proteins, and these gap proteins help the Hras become active as a GTPase to hydrolyze Hras to GDP, and therefore changing the Hras structure and therefore turning Hras off. Okay, so that's how Hras is inactivated, through its own GTPase activity that's enhanced by the gap proteins. Now, how does Hras become activated? Okay, how is this GDP converted to GTP? Well, actually, it's not a conversion of GDP to GTP. It's an exchange. So what happens is the GDP is physically pushed out of the Hras molecule. It physically leaves, and within the cytosol there's a much higher concentration of GTP than there is GDP. So once you displace GDP from the Hras, it's typically replaced by a GTP, a triphosphate, returning Hras into the active form. So to go from inactive Hras to an active Hras, you need to physically remove the GDP through a process called nucleotide exchange, and then this is replaced by the GTP from the cytosol to reactivate Hras and cause that switch region to that effector loop to change conformation and then signal to the cells. And just for the sake of completion, I've kind of included here that there's probably an empty Hras structure involved in this cycle as the nucleotide is displaced and then the new one is picked up. But effectively, Hras inactivation occurs through hydrolysis, and Hras activation occurs through exchange. Okay, so hydrolysis, nucleotide exchange, hydrolysis, nucleotide exchange. Okay, and I've shown here that the gap proteins facilitate the hydrolysis. There are also other proteins which are helper proteins in the Hras cycle, and these are referred to as guanine exchange factors, or GEF proteins. So these guanine exchange factors are other proteins that help displace the GDP so that GTP can replace it. And these are these guanine exchange factors. So this is a slide taken from another one of these lectures in the series. This one I think came from the cell signaling or cancer signaling presentation. And we covered one of these linear signaling pathways from the plasma membrane through to the nucleus to activate gene expression. And an important component in that signaling pathway was the Hras protein here. And we talked about how the SOS protein activates the Hras protein. So the SOS protein, it turns out, is a guanine exchange factor. So the SOS protein has the ability to help the Hras protein to displace the GDP in an inactive form, and then to pick up the GTP and turn Hras into an active form, and then the effector loop can act with one of these effector proteins to then signal to gene expression in the nucleus through the MAP kinase pathway. So the SOS protein is a guanine exchange factor, that provokes nucleotide exchange in the Hras protein. So that's part of the on switch to activate Hras. And as we've already mentioned, there are other protein activities called GTPase activating proteins. These are negative regulators of Hras. They help the Hras protein substantially be more effective at hydrolyzing the GTP, and therefore this makes the Hras inactive, and therefore the GTPases act as negative regulators. And I always find this confusing in the textbook because it's referred to as an activating protein, but the activating protein is actually a negative regulator of Hras. So the GTPase activating protein turns the Hras off because it makes the GTPase of Hras more active. The GTPase hydrolyzes GTP to GDP, and therefore Hras becomes inactive. So if you think about it, it's logical. If you just associate the word activating with Hras, then you're going to get confused. Okay, so the GTPase activating protein activates the GTPase, which turns off Hras signaling because the GTP is converted to GDP, leading to an inactive Hras. Okay, so hopefully that's been helpful to you, having a look at an oncogene and looking at how an oncogene can function in a signaling pathway. And thank you for listening.